Hi, everyone. Welcome to episode 178 of the Tick Bootcamp podcast. The title of today's interview is Targeted Treatment, an interview with Dr. Alexander Shope and Gary Shope of Contamination Source Identification. My name is Richard Johannesson.
And I'm Matt Sabatello.
Matt, one of the worst things about the Lyme disease experience is that the Lyme disease testing just sucks. People are unfortunately on these long journeys because doctors don't have the tools they need to determine, at least in their minds, whether or not someone's suffering from Lyme disease or not. And now we may have found a group of people who are going to change that problem.
And Rich, what always makes me weary about new technology is that the Lyme wars get in the way and these companies get deterred. But Dr. Shope and Gary will not be deterred. They have a long career in advancing medical technologies into the marketplace. And Gary assured us he's going to push this through and will have this testing available within the next year.
So, Matt, I think it's really exciting that we have something called untargeted testing for targeted Lyme disease treatment. And without further ado, I'm really excited to introduce Contamination Source Identification with Dr. Alexander Shope and Gary Shope. Hey, Gary and Alex, and welcome to the podcast.
Thank you, Rich. Good to be here.
Yeah. We are really excited to have you guys. I want you to know that you are a part of a very select group of people who do not have Lyme disease that have who have made it onto our podcast. So welcome non-Lymies to the podcast.
Well, thank you. I'm happy to be in that category, actually, based on everything I've learned.
Yes, me too. So the reason we invited you folks to come onto the podcast today is because we've been doing a series of podcast episodes on testing. And it's one of the places where we think there needs to be a substantial change. There are people who have been on diagnostic journeys for 20 or 30 years in some cases because the diagnostic testing has been so poor. So we want to first introduce you, Gary, to the podcast audience. Can you please share with our audience your background?
Yes, I'm basically, people refer to me sometimes as a serial entrepreneur, although I don't think I deserve that category, but I've been in the medical device business for about 40 plus years, mainly in the cardiovascular and orthopedic businesses. And a few years back, I sold a company to, I sold an innovative hemostatic agent to a big company and retired. And then I came out of retirement and decided to get back in the saddle and created actually one company that is an antimicrobial technology. And and I've created a spinoff now called Contamination Source Identification that is a molecular biology diagnostic lab. So I've been doing this for a long time, but like I said, I just came out of retirement to do this.
Well, welcome back. So, Alex, can you please introduce yourself to our audience and share your background?
Yeah, sure, Rich. So I'm Dr. Alexander Shope. My background, I'm obviously a physician. I went to Penn State's College of Medicine. After that, actually, I went and spent years doing a research fellowship at Rothman Orthopedics in Philadelphia, where I was working with Dr. Javad Parvizi, who is sort of the thought leader and key opinion leader on infected total joints, and really just kind of sank my teeth into some more of the infectious disease diagnostics there with him, and learned about some of these new innovative techniques. And sort of while there, my father was kind of starting this journey with CSI. I introduced CSI to Dr. Parvizi, and then ran the studies that we did alongside with CSI, and got really interested in it. And frankly, I've always been interested in business and innovation, and made the decision that this was going to be my career path, and hopped out of clinical medicine so that I could pursue this and help bring this both our companies actually to the market.
So, Gary, let's talk about the entrepreneurial model. As a serial entrepreneur, I'd like you to talk to us about why you believe the best model for helping folks in the Lyme disease community is the entrepreneurial model rather than an academic model or a not-for-profit model or some other model.
Well, that's a great question, because what's interesting about this, our company is in central Pennsylvania, a little town called Huntington, Pennsylvania. And actually, when we first did this, when Dr. Parvizi first challenged us about proving our worthiness in this diagnostic capabilities. He literally said, "Are there roads that go into Huntington, Pennsylvania?" We're right over the mountain from Penn State, but I assured him that Federal Express delivered at 10:30 in the morning in Huntington, Pennsylvania, just like they did in Philadelphia, so that our clinical samples could get there. But the entrepreneurial model is good because there are many issues involved, but being able to change on a dime is, and being able, and for instance, our changing from our focus on orthopedic and sepsis and other unmet needs in clinical medicine, and quickly changing to Lyme because and tick-borne diseases because we recognize the dramatic unmet need, is something that doesn't happen in a big corporation. And also being untethered to a large academic institution and all the layers and structure that hold you back. I became, I'm not going to name the institutions. I was actually trying to create a business whereby we could bring the technologies that are captured in these medical academic institutions and bring them to the marketplace. And that's not easily done because of all the layers of consideration in those places. So my experience has been that small companies can recognize an unmet need and can execute. Our goal is actually to spin the company back to a larger entity that can really take advantage and bring it to the broader audience, both in the US and the United States. But the startup process and our ability to work with the scientists and get things moving in the right direction without having to go through meeting after meeting is important. I don't know if that answers your question, but that's
it does. I appreciate that answer, and I really like it. So, Gary, before we move on, I actually have to ask your permission for something. I was recently slapped across my hand with a ruler by one of our followers because I referred to a doctor guest as doctor, and I referred to the non-doctor guest by their first name. Do you have any objection to me referring to your son as Dr. Shope and you as Gary during the rest of this podcast?
No, I do not. His mother has made it clear that that's the way it should be.
Okay, so Dr. Shope, am I pronouncing your last name correctly? Is it Shope?
Shope, yes.
Oh yeah.
So, Dr. Shope, please share with us why you chose to move into the entrepreneurial model and you moved away from either the traditional hospital model or an academic model for doing your work.
Well, so growing up, actually, with my father being in the kind of medical device field, and obviously then being an entrepreneur himself, and just watching everything he did. Well, funny stories I used to tell him when I was younger. I just knew I didn't want to do what he did, and here I am doing the exact same thing. But he naturally had a lot of physician friends. Many of them, as medicine has changed throughout the years and become more health system based, had left clinical medicine and gone on to do things in industry. And that always just intrigued me so much because I saw myself as a physician owning my own practice, being able to kind of have more control over the business side of things. And that's just not how medicine is anymore. It's all large health systems. It's all really becoming, these MBAs and executives telling the doctors how to, see their patients, how many they have to see. And I really took issue with that and just didn't like that type of a model. But one of my father's good friends, Dr. James Hart, who's a just fabulous cardiothoracic surgeon. He's now retired. He had left clinical medicine and gone into industry, worked for Johnson & Johnson, and was their CMO actually for their Ethicon division, I believe, before retiring. But he said something really interesting to me one time that always resonates, and it's what I live by now. He could have seen, 30 patients a day, operated on, a couple thousand a year, change those lives. And that's great. That's fantastic. But by stepping away from that type of medicine and becoming this more, entrepreneurial thing and driving a new technology to market, you can impact millions of people by having something like that. And I truly think we have a technology that's going to be able to do that here in the long term. So that's sort of been my journey and what pushed me in this direction.
And I want you to pause on that technology for a moment because I need to get a little more background from Gary. So Gary, talk to us about CSI and the type of work CSI has done other than in the Lyme arena? And we'll get to Lyme in a minute with Dr. Shope.
Well, actually, CSI's focus and business focus wasn't originally Lyme. As I mentioned, it was an afterthought. But our business, we've created a platform in infectious disease diagnostics that nobody else in the world is doing right now. So the scientists, including Alex, that my co-founders, Dr. Regina Lamodella and Justin Wright, have figured out how to analyze RNA sequencing in such a short period of time. So we were focused on sepsis and prosthetic joints and post-cardiac procedure infections and also all those unmet needs. So our business focus is we still believe that is going to drive us to the acquisition phase so that a bigger company will be interested. Also, the value proposition, now, and this is sort of inside information, but reality is the value proposition of CSI is actually the bioinformatics, or the amount of information we gather. I mean, we were looking at COVID variants last year because we're RNA sequencing. We do whole genome RNA sequencing. So we see the entire pathogen, so to speak, so we can definitively define it. But the bioinformatics and the updating of our database and the patents that we have on how we handle that information will grow and grow and grow. And in the future, when they're looking to create new antibiotics, new vaccines, they're going to want to know what the latest mutations is for all pathogens, not just bacteria for antibiotic resistance, but viruses. And in the case of tick-borne diseases and Lyme disease, we don't know at this particular point what's happening to the bugs with regard to mutating and maybe warding off the antibiotic therapy that they're given. So our direction is to create the platform that analyzes that and takes that forward for the future as well as the present. The diagnostic capability is good because it treats patients, or it defines disease states today. But the future of artificial intelligence and predictive medicine and precision medicine is really what we're all about.
Okay, so Dr. Shope, you now have to interpret that for us, because that was very powerful, but certainly way over my head. So Dr. Shope, tell me about what is different about what you folks are doing there, and sort of unpack what your dad just outlined for us.
Happy to do that. It's funny. It's usually opposite. I'm the one that says those complex things, and then he yells at me and tells me I have to be more simple or something. So this is funny. Shoes on the other foot today. So what makes us very different from everybody else? Culturing was always sort of your gold standard, growing things in petri dishes. And that takes, several days, and it was an inefficient method. Now, there are new methods out there that are trying to harness the power of DNA, because everybody knows about Ancestry, DNA, 23andMe, swabbing cheeks, getting sequencing data back that says, hey, here's where I'm from. There are other techniques that are they're targeted. They focus on a very small gene region. It's kind of like a fishing hook. They go looking for a small gene segment that is associated with a pathogen, and they run these panels. That has problems because maybe an infection or something's being caused by something that's not on your panel. You don't have the right hook or the right bait. So the natural next step beyond that is doing something that's untargeted, which is what we do. We don't focus at all on a specific gene. We just sequence everything that's there. Furthermore, the other problem with DNA is that it doesn't indicate whether something's living or dead. You could find DNA on dinosaur bones and find remnants of it. It's a stable molecule. RNA, on the other hand, is an exact copy of the DNA, but it's very unstable because of its just molecular nature. After a cell dies, it's only around for about 30 or 40 seconds at most. So if you can identify something based off of an RNA signature in this untargeted manner without fishing hooks and things, you have a much better chance and better understanding of what's truly active and causing an infection. We sometimes liken our test to fishing with dynamite instead of hooks, if we want to stick in the same type of analogy framework there.
Dr. Shope, before we go into the different type of testing modalities today in the Lyme world, I want to ask you about the difference between RNA and DNA, because we've learned from past podcast guests that DNA and the dead Lyme bacteria can actually have a negative impact on your health as well. So it sounds like you guys focus more on the RNA, but is there an implication that DNA can have in keeping people sick as well with chronic Lyme and tick-borne illnesses?
So I know that is pretty widely debated, and there's also a bit of a debate about how long the DNA can circulate within your system. Does your body clear it? Does it get broken down by other proteins called DNases? So I don't think that there is a specific and definitive answer. I think that it potentially could. I recently read a paper where they were finding some antigens or leftover proteins from the Borrelia species, embedded into the the synovium of the patient's joints, and they think that could be causing some inflammatory reactions. So there is a chance that some of these dead bacteria and what's left over from them can cause some aspects of chronic Lyme. But the actual nice thing, and this gets a little complex about our technology, we are RNA sequencing, but in the nature of how we do that method, we actually are able to analyze some of the DNA as well, but we then take it from a computer processing standpoint and can remove certain aspects of that and then focus in on the RNA. So we do harness some of that DNA data if it is there, but the RNA data is what's really highlighted then.
So before we go into the details about your specific testing that you're working on, can you talk to us generally about the current Lyme tests that exist and other tick-borne illnesses and why they're flawed in different areas? So for example, talk to us about the traditional ELISA, the Western blot, and then what's being done by some of these big Lyme testing labs.
Absolutely. So let's start right with the ELISA and Western blotting method, which for whatever reason is still the gold standard because it is quite flawed. So what that is doing is simply looking for your immune system's reaction to a pathogen. Your body naturally produces antibodies when it sees something that's not supposed to be there. The thought process then being, okay, if your body's reacting to a Borrelia species or to some type of tick-borne pathogen, it's eventually going to produce antibodies. So if you detect the antibodies, therefore you should be sick. The problem is that the secondary test they use, the Western blot, create these little banding patterns. It looks for specific proteins, and they've created a pattern that says, oh, you have to have so many of those bands before you're positive. And I'm sure sure the listeners here probably know much of that already. The issue with that is it's outdated. There are certain periods of time that it takes your body to build up the antibody response to the bacteria. So you might not be positive right away or within a certain time frame. But furthermore, there are certain bacteria that don't necessarily create those proper banding patterns on the Western blot. So there are multiple levels about why that is just a flawed test. It's also not a direct detection of the pathogen too. So it used to be the best way of doing it, but we have much better technologies out there now to directly detect these types of pathogens that really can hide from the old culturing methods.
Dr. Shope, I'm sorry to interrupt, but before we go on to another testing method, can you talk to the fact of that there are different strains of the Lyme bacteria, and these Western blots and the ELISA only look for certain strains, and also what impact a chronic Lyme patient would have on these testing if they're immunocompromised?
Certainly. So, all bacteria have different strains, and the strain being just a different genetic variant of them. When their genetics change, typically that changes some different structures within the bacteria itself. It could be the outer proteins they express, it could be some of their internal mechanisms. So some of these different strains and different species of the same, genus, the Borrelia genus, might have different proteins that would not get tagged on that Western blots the same way as as what the original Borrelia burgdorferi did. Even the Borrelia burgdorferi, because everything mutates and changes over time, that's natural selection. It's if you don't update your diagnostic criteria to the banding patterns you see or expand it, you're not going to follow what's happening from an evolutionary standpoint, and therefore it's going to eventually evade the detection method.
So now that we understand,
Uh,
and I think most of our listeners understand that the current Lyme testing, at least in the Western blot, is just god awful. Can you tell us a little bit more about these other tests that are available that are recognized in the chronic Lyme community, but also why they may not be as perfect as we think they are as well?
Certainly. So I know some of the other newer techniques and things too, whether it be, some have used different criteria. They expand upon the different banding patterns. Like I said, they also, look for numerous other pathogens as well. One of the things that I see commonly, especially within other infectious diseases, would be a test called a PCR test. It stands for polymerase chain reaction, which gets technical, but essentially that's the fishing with hooks type things that I had mentioned earlier. They're taking known segments of these bacteria's genes that are in, their DNA, and they say, hey, this segment is associated with this bacteria or this particular strain of bacteria, and they create this little panel. So they go in and they get these little primers that will latch on and then get amplified, and that creates the detection. But again, the problem with that is if they're not looking for it, if they don't have a particular primer for a specific species, they'll miss it. Again, then we run into the same problem with the ELISA and the Western blots, where I mentioned the evolution of the bacteria. That DNA changes over time. And so at some point, those primers could be missing things as the bacteria rapidly change and evolve. And, and then those primers won't work. So again, that can be a problem, let alone that, if the bacteria is actually potentially dead, the primers will pick that up, but maybe it's the living organism that they're not detecting.
So Gary, let's take this back to the introduction you gave us a moment ago, which was you're using AI and other tools to now look for a broader spectrum rather than having the metaphor that your son had used, the fishing hook to hook just one singular fish. Talk to us about how you're doing it differently than they would be doing it with a traditional PCR test.
Well, actually, when I was talking about AI and predictive medicine, we're actually that's more of a futuristic view. Our lab is the only that we're aware of, the only vertically integrated lab, in that we do all of, even though we're just a small company in central Pennsylvania, we're the only lab that does our own wet lab work, our own sequencing work, and I referred to our bioinformatics. We have our own supercomputer on site where all that information and we've invested in what's called a laboratory information management system, because we found out that one of the problems of labs is that they lose samples all the time in reporting systems. So we can dump the information directly into electronic medical records.
Okay, so let's pause there for a second. Tell us why all of that would be significant to somebody who is sending a test sample to you as opposed to another lab. Why would you be better?
Well, and because we're faster and more complete, and I think the answer to because I was going to say that we have patents on the wet lab work and patents on the bioinformatics, and I can't get deep into the patents in this conversation with regard to the wet lab work, but
we understand that. But just let's talk about it on a high level. I get bitten by a tick. I decide that I want to work with your lab. I send my urine sample to you. Why would you be better just from a procedural standpoint than me working with another lab?
Because what our test procedure does is that it immediately recognizes within 24 hours of receipt of sample, whether, and that applies to sepsis and all these other things. We can see exactly what is living. We created a urine test to make it easier on the patient, even though we can see it in blood as well. But we see everything that's living in the urine, and not just the one or two bugs that somebody else may be looking for. And what used to take companies weeks to analyze for that kind of complex analysis, we can now see in minutes. And
okay, so I'm now sending my sample to your lab, and your son was just outlining for us the challenges with traditional PCR testing, which is one hook, one pathogen, right? If we use your lab for my urine, are you telling me that we're not going to have one hook looking for one pathogen, but there's going to be a whole broad spectrum of pathogens that are going to be evaluated and provided to me and my physician in a very short period of time.
Exactly. Exactly. We're going to see everything that is alive. Our what's called our level of detection that has been validated is that we can see as few as three pathogens, three bugs per milliliter of urine. That is off the charts with regard to being able to see what's there. So, to be clinically relevant, I guess, in some of these other tests, I guess you have to see somewhere in the vicinity of 50 to 80 bugs before somebody can define it. But we can see just a minute number and be able to define. But now, interesting enough, Alex was talking about PCR testing. We're working with a company that gathers ticks. When you get bit, you can send it in, and they're working with us because this collaboration is really important. And they use a PCR method, and they're opening up these ticks and reporting back, and they've been able to see like four or five different pathogens in these ticks, and we've seen a lot of different pathogens in these same ticks because our testing is so specific. So I think one of the issues is with regard to patients, and we don't know this. I'm going to talk off the cuff here, but why is it that some of these patients react to antibiotic therapy and others don't? And we've learned a lot from COVID, for instance. If I can digress a little bit, we've watched and heard in the news how different people are reacting different to the COVID virus. And it has a lot to do with your disease, your immune system and other disease states you may have on board. Well, isn't it possible that the same thing? There are people that are reacting differently to the Lyme bugs and the different pathogens that are showing up, and we don't know why. But the fact of the matter is, if we're seeing everything that may be impacting those chronic Lyme patients, it's going to be a helpful process with regard to problem solving which antibiotics to use, because we don't even know that the antibiotics that are currently used today for Lyme patients are being effective for the broad spectrum of pathogens that we're seeing in the urine. So it's a lot of research to be done with regard to the effect of the treatment. And if I could take one step further, you treat somebody today with an antibiotic, how do you know that it's cleared? How do you know that it's done? And so that ongoing surveillance and being able to specifically say, yes, it's gone. The living pathogens are no longer there living, or no, it's not gone. So we can define the treatment therapy.
So Gary, before we move back, because I know Matt has some questions to ask Dr. Shope, who, by the way, you referred to as Alex by mistake, I think a moment ago. There is some research that is suggesting that at least the black-legged tick could carry as many as 200 different pathogens. Yet we're currently only testing for, at most, I think, 7 or 8 different pathogens. So can you talk a little bit about how your lab is sort of changing the conversation in that regard? Because viral load is certainly going to be significant to just sort of, to tack on to your conversation about COVID, where we know that people were getting sicker when they had a larger viral load, in addition to those other factors that you outlined. Talk to us about what you're finding with the ticks that you're examining and how it's different from the traditional PCR tools that are being used.
Well, I'm actually, if you don't mind, bouncing that over to the good Dr. Shope here, because actually he has a slide in some of his presentations for physicians where we can show the diversity. And Alex, I actually don't even know the number of additional pathogens we're finding in the live ticks, or the ticks that are being sent to us.
Yeah. So I think we've received so far, just this is hot off the press actually. We received 7 ticks. The PCR method identified regular Borrelia burgdorferi in 6 of 7, as well as then a Babesia species and an anaplasma. So they saw 3 pathogens total spread across 7 ticks. We did our analysis on those exact same 7 ticks. We identified the exact same signatures of those 3 pathogens, and then we also saw an additional, I want to say it was 15 or 20 other pathogens.
So that is very significant in in the tick-borne illness community. But Dr. Shope, I do want to expand upon what we were talking about earlier as far as running these tests for Lyme patients, specifically people that are sick with chronic Lyme, and not just looking for Lyme disease and not just looking for several strains of Lyme disease, but also looking for these other co-infections like Anaplasma, Babesia, Ehrlichia, Rocky Mountain Spotted Fever, and beyond that, other things like parasites that we know ticks can spit into humans, and also viruses we can get from ticks as well. So what breadth of testing are you doing beyond bacteria and Lyme in your testing compared to the ones that are on the market today in the the top best testing facilities that we have available today for Lyme disease?
So again, the unique thing about this RNA-based method is that we actually test for everything all at once. So we're not just looking for bacteria. We're not just looking for viruses. We look for everything all at once, as we're capable of seeing all bacteria, fungi, viruses, parasites, other eukaryotic organisms. So kind of all the above, which is, again, what is so great. It's untargeted, and we can see everything that's potentially causing somebody to be sick, including those, the co-infections. We can, catch, the Babesia. We can catch if somebody has, Anaplasma and Rickettsia, that we see it all at once.
Yeah, and I think that's, if I might interrupt there a little bit, that I don't know that that's the uniqueness of what we bring. What do we bring? The difference is with one test, we see all of it. We're not looking to validate anybody having just Borrelia. We see whatever's there. We see it with one test.
So talk to us a little bit more about mutations as well, because I understand that your testing not only can find currently identified viruses, pathogens, etc. It also can find mutations that aren't even known yet. Is that correct?
Yes. Yeah, because we can utilize the sequencing data to identify those variants or the mutations within the genes and the DNA and RNA itself, and then utilizing our database in the bioinformatics with machine learning, we can also potentially predict when certain mutations and other things will pop up. But again, it's sort of like with this COVID pandemic that we all went through. We could potentially, ping something like that, where there's all of a sudden like a mutation in something that was, benign before, that we've, seen it was benign, but we all of a sudden pick up on a mutation is causing an illness. Because of the nature of RNA in our untargeted testing, we can catch things like that, and we can just see them right away, which is what is so unique.
And the antibiotic resistance as well.
Yes, that's it.
Yeah, so talk to us more about the antibiotic resistance. So how is that possible? What are you doing to identify what pathogens are resistant to what antibiotics, and how is that information meaningful to doctors and patients?
So we always are looking for antibiotic sensitivities for the pathogens that are causing infections. With in the hospitals, when it was culture based, you were waiting on, they would use little antibiotic discs to see if it would kill the colonies that were growing on the petri dishes. Stepping into the molecular world, your DNA is essentially just a recipe book. It has instructions for everything that your cells need to carry out their lives and their functions. Therefore, if something's resistant to an antibiotic, it's got the instructions built into its DNA. But again, we don't always use all the recipes in your recipe book or in your DNA. So it might have the potential to be resistant, but it might not be actively expressing that. RNA, though, if there's RNA, the bacteria is actively trying to express that gene. It's trying to avoid that antibiotic that is associated with that sequence data. And again, gets very data heavy and things, but because we can, if we see an antibiotic resistance gene from an RNA signature, that bacteria is trying to express that, and therefore maybe a different antibiotic is the way to go. And that's very important information, just within the infectious disease community as a whole, but also within the tick-borne pathogens as well.
So Gary, give us the layperson's analysis on how your tools are going to allow doctors to identify strains of a pathogen that are going to be resistant to antibiotics.
Well, if if we could just draw a parallel path here with sepsis and tick-borne diseases a little bit. I think maybe some of this is understandable because the same thing occurs in both disease states. Before COVID, more patients died of sepsis in a hospital than anything. 1 in 3 patients who died in a hospital died as a result of sepsis. That's statistically, that's what's been published. And I learned from the good Dr. Shope that it's well published that for every hour that a sepsis patient doesn't receive the right antibiotic, the mortality rate goes up by 8% per hour. And the physicians are there not knowing what the bug is. So, and again, the same thing occurs with as far as I'm concerned, with regard to chronic Lyme patients. You don't know why that patient continues to be sick, because it could be some co-infection or some lingering pathogen. Our ability to identify the living pathogen that is replicating, again, the RNA, it tells you what's living. And when all my young scientists, all these young kids that surround me, all these bright kids, I don't know what they're talking about half the time, but in layman's terms, what they're able to see is the pump mechanism, for instance, that the antibodies coming in, the bugs learn to either, when you're developing a pharmaceutical, you're developing one mechanism of action, and pathogens, including the pathogens that, as it pertains to tick-borne and Lyme disease, create mechanisms to they mutate so that antibiotic doesn't affect them. Well, the RNA sequencing is able to see what it's doing in its mutation to cause that to happen. That's what Alex talks about, its expression. And so therefore we're able to identify that expression. And in the case of sepsis, and we're assuming when we publish in the case of tick-borne disease, we'll be able to tell the doctors that maybe what's happening here is the normal antibiotic that you give isn't being effective, and this thing is not being killed because it's created a antibiotic resistance mechanism to push it away.
So now, how are you able to see that the particular pathogen is resistant to the antibiotic that has been prescribed by the doctor? I'm still not clear on how you can do that.
Alex, do you have a layman's version of what you guys do in that complex world of yours?
Or some metaphor?
Let me, I'm thinking about a metaphor, the best way to explain it. Well, we are able to associate from our big supercomputer. We'll pick up the gene sequences associated with the pump or the mechanism that allows them to avoid it. So if they're basically slingshotting the antibiotic back out of their system as it comes in, that's their mechanism. They're building that slingshot based off of a set of instructions, which is essentially the RNA. If we see that RNA set of instructions from our analysis, we can say, hey, don't use the antibiotic that they can slingshot back out of their system. Use this little hand grenade that they're not going to see coming, and allow that to kill the bug. How was that analogy?
That was a very good analogy. So Dr. Shope, as an IT professional, I'm really intrigued by the fact you keep talking about big data and data processing and consuming large quantities of data and interpreting that to provide useful information to doctors and patients. So talk to us a little bit more about this supercomputer that you have in-house and what algorithms that you guys have built to kind of identify not only what antibiotic resistance these different pathogens have, but also what type of pathogens and bacteria and viruses that you're finding in your test results as well.
Right. Well, I mean, I can talk a little bit about that, but that isn't necessarily my area of expertise. So it's what we call our rapid DX bioinformatic pipeline, and it's really more our business partner Justin Wright's really like brilliant work and area of expertise there. We have some patent submissions surrounding this, so I can't again get too in depth into it. I'll let you know that it's called Rapid DX. Again, we just think we like fun acronyms like that. Essentially, it is just rapid parallel pathing of the data analysis. And frankly, that's all the more I really understand of it. But being able to process that much data so quickly and then pumping it back out into the electronic medical records and making an interpretable report is such a key thing within medicine, and it's going to be so helpful to the physicians who are interpreting the data on their end, as well as to the patients who are then receiving care.
Well,
Not sure I really was able to answer your question too well there, but
well, Matt, the
I
one
hope I
thing.
answered parts.
Oh, I'm sorry, Matt. The one thing that maybe your audience doesn't realize, that even though we're making this sound very simplistic, when we go through this process of receiving the sample and then we do the sequencing, we get all this information of the genomic makeup of the pathogens. There are hundreds of millions of pieces of information being spit out that goes into this analysis that quickly, like I said, used to take them weeks to do what they can do in minutes with what, and the systematic approach that we have presently. Even we're a small company, we have presently, I think, four folks that do nothing except focus on this whole bioinformatics side, this analysis of what is the antibiotic resistance expression and everything in this database that we use. And obviously the value of these ticks we're receiving, because we plug all that information as a reference library. So the next time we see it, we can recognize it and update its mutations. But there is hundreds of millions of pieces of data being fed into this thing and spitting out one single answer with regard to what's going on in that patient.
So Gary, let's go back to the ticks that you're using as now a reference tool. Are you finding pathogens in the ticks that have not been identified by other researchers yet? And are you feeding that into your system for the comparative purposes that you're using them?
I'll start the conversation. I hope Dr. Shope finishes the conversation, because actually and the answer to your question is yes. We use all that information to update, and every time our computer system sees a variant, whether it's COVID or whether it's Lyme or tick-borne, it updates it, and that's what we call our living database. That's the value proposition. And you can imagine 4 or 5 years from now, when they're saying, "Hey, I wonder what's out there," they can look at our reference library with regard to all the mutations that we've seen over these 4 or 5 years and what's present today. But even in our last year, in our initial analysis of some samples that we received from central Pennsylvania, we saw not only did we see samples of Borrelia that's only supposed to be in Europe, but we found it in central Pennsylvania, but we did see unidentified Borrelia. So that variant is now plugged into our library. So it will be seen and referenced the next time that comes through our scope of our view.
Now, the tick testing is really important, Dr. Shope, because I'll use myself as an example. I was bitten by a tick about 5 weeks ago. We sent the tick out for tick testing, and the tick came back clean, meaning none of the traditional pathogens that are looked for with a PCR test were located in the tick that had bitten me. Now, I decided to treat anyway because I believe that it's likely that the tick that had bitten me had pathogens that were spit into me that were not identifiable with the traditional PCR test. Are you finding now that the ticks that you're testing are harboring pathogens that are not traditionally identified in the 8 or 9 or 10 pathogens that are being tested with the traditional PCR test?
Yes, the ones that we've tested so far, there were absolutely a number of different Borrelia and rickettsial species that I know are not traditionally tested for on regular PCR and tick-borne panels.
Anything else? Now, my understanding is that what you folks have the ability to do is test a sample, strip out all the human DNA, and then see what's left. Is that the way you guys do this? And if that's the way you're doing this, do you believe you'll be able to identify other pathogens that ticks are spitting into us that are not currently being identified?
Oh, yes, absolutely. And that was a very good, just kind of overview of one of the things that we're able to do as far as the stripping away and focusing on that very small pathogenic signal that we have in us. But yes, because we're untargeted, we'll be able to see anything that is there. And we are not panel based. So if it's a new thing that's not typically tested for, we'll still see it if the sequences are there.
Yeah, there's a word of caution though to those who are listening that because we've just the tick is a dangerous little bug, but the one thing we don't know as we view all these pathogens, we don't know, just as is the same thing is true when we deal with hospital acquired infections. You have a whole list of bugs that actually CSI is actually doing a lot of environmental testing in ICUs and hospitals these days. There's in conjunction with, that's how CSI got started, working with this other company doing surface environmental testing in ICU hospitals. But you get this, we see this whole list of bugs in a hospital, but only a very small number of them really cause the hospital acquired infections. And the one caution with regard to a tick biting, it may introduce all those things, but at this present time, we don't know which one, and each individual might have a different reaction to all those pathogens that are being introduced. So which one is causing the response or the disease state that the patient is experiencing, we don't really know, but we will see them if they're there. That's the important thing.
Well, but Gary, we can't even identify them right now, right? I'll go back to my example of my tick bite from 5 weeks ago, right? I don't know what combination of pathogens were spit into me. I just know that those which are traditionally tested were not spit into me, at least with the PCR test that was done, right? So we can spend a lot of time talking about first identifying what is spit into us versus what's not spit into us, but we can also now identify combinations because we recognize that let's put aside the differences that each one of us have physiologically, but we also know that the combination of a multi-germ infection is going to be different because of the combination, and we don't necessarily have the ability to determine what the combination is if we are using some test other than the system you appear to be using, which is to strip out the human DNA, and now we can see everything in there, right? So one of the things we recently learned from an interview we did with Dr. Alan McDonald is that there are many pathogens that are being spit into us that are not bacteria, that are not viral, that are not necessarily parasitical, parasitic, that are also causing us to get sick. And it seems to me that the only way that we could identify those pathogens as potentially causing us to get sick, Gary, would be to strip out the human DNA and see what else is left. Or am I wrong?
No, that's exactly what I do. You know, I'm getting a chuckle the fact that we've invented a new term for the introduction of pathogens. The bugs are spitting them into us now. I got no because, but no,
that's what they do. Yeah.
You know, what you're describing actually, we keep circling the wagons here, but what you just described is exactly what we do. And when we use the terms untargeted whole genome sequencing, which means that. And actually, Dr. Shope always used, when we're talking prosthetic joint or sepsis, he always, actually the secret sauce of what we do with regard to blood, urine, synovial fluid, cerebrospinal fluid is exactly that. The reason that all these other computational methods are slow with regard to what we're doing is because all that human RNA signal is there. And what our folks have figured out how to do, and Alex always says it's like looking for a needle in a haystack. What we figured out how to do is to burn the haystack and walk in and pick up the needle. So all the needles of the pathogens that the tick has introduced into you, that we will burn that haystack and see every single needle that is in your urine or in your blood, because we just figure it's easier for the patients to give us urine samples versus blood samples.
So, Dr. Shope, I know that what may make me sick may not make you sick. And I think we sort of learned that with Tickman Dan that we had on the presentation earlier in the week, where he's been bitten by ticks hundreds of times and never got sick, right? So clearly there's something unique in his situation that's helping him stay healthy, whereas something in my situation caused me to get chronically ill from various tick bites. So I think compiling all the information that we can get after what's left from stripping out the human part of the RNA and leaving all of the pathogens, that information is going to be super helpful because we're all uniquely different in regards to what may make us sick individually. Is that something that you would agree with from a medical standpoint?
Yes, absolutely. That was very well said. I think that is very key because we are all different, and our immune systems and the various factors, that make up who we are in our physiology, are different. And Thickman Dan, who's I forget how many times he said that he's pulled a tick off himself. It was pretty astounding how many, though, and he's never had Lyme disease or anything. But yeah, the fact is he might have some sort of a protective mechanism that maybe you or I just don't inherently have. And compiling all of that data along with, as we continue to study this and having the patient's data, their disease history, their clinical symptoms and manifestations, in addition to all of the pathogenic data that we are generating from the actual samples themselves, feeding that into the machine learning algorithms, which I don't personally completely understand, but using that with the machine learning process to better understand maybe who's more susceptible to these types of infections and all. I mean, that's all things that will be done in the future, with this type of research and all. But I think that could be very important with the direction that medicine is going.
And I think all of this wouldn't be possible without technology. So at a different level, LymeDisease.org has maintained this My Lyme Data Registry, which is a patient-powered data machine that provides information such as what's the most common symptom for chronic Lyme disease, what's the most effective treatment for chronic Lyme disease, and they're providing it to Lyme patients so it can help them make informed decisions for their treatment and also for their diagnosis. But I think what we're talking about here is so much more powerful, and it's taking it to just the complete next level of building a database that you're going to be able to identify all of these pathogens. And then once you get your testing out there and you're just building this machine and you're using big data, you're going to be able to hopefully identify which of these pathogens are more severe than others. And then down the road, maybe 3, 4, 5 years from now, be able to sort of chart that for people to say, here are the pathogens you have. Here are the ones we're finding to be the least likely to make you sick. And here are the ones that we're finding to make you most likely to be sick. And that data can be super powerful for physicians and patients and hopefully eradicate a lot of the suffering that we're seeing in Lyme patients today.
Absolutely. And that is really the hope of, that is what we hope we can really help and do. Well, that's what we think it's what we know we can do, frankly. But, we're in the first steps of it now, and it's going to, take some work and all, but we're really excited to be able to, bring this forward, because this is truly what, just the tick-borne community needs, is this type of testing.
Yeah, Matt, there are actually 2 stages. Our first goal is to allow infectious disease docs to understand that these chronic Lyme patients and acute patients, but particularly the chronic Lyme patients, do in fact indeed have pathogens in their body. So that's the first definition. Long term, all the information we give them on the variety of pathogens that might be there. I mean, that's obviously a secondary goal, because because you got to worry about, okay, what therapy needs to be applied to properly attack that. That's a secondary goal for future years here as well. And the third goal from our perspective, you think about what we do with regard to recognizing antibiotic resistance. We use the term, and Dr. Shope uses the term, this is his term, precision medicine. I mean, it's sort of a buzzword that's thrown around, but what that means is that by just taking an antibiotic, that's, and sepsis, whether it's sepsis or Lyme disease, just by arbitrarily taking antibiotics because you think it's going to help, that creates antibiotic resistance in the bugs. They continue to mutate around that because it's not effective therapy. But precision medicine means that by identifying the antibiotic resistance characteristics, then you change the antibiotic so that it's targeted so that you can kill that pathogen right away by using the right antibiotic. Those are critical factors as we go forward. Those are the problems that we want to help solve and create more research to help solve those problems going forward, because that's what's going to help patients.
So Gary, from a business modeling standpoint, I'm wondering whether I can suggest to you that perhaps you open up an element of your business where you're not just receiving ticks from one lab, for example, but that you become a tick testing lab yourself. Because if you have people who are purchasing your services, your tick testing services, they are going to be getting a substantially better report than anyone else is otherwise able to provide. But at the same time, you're also now building up this database where you're identifying all the non-human DNA and RNA that's in these ticks. And you sort of have the best of both worlds. And then again, I'm going to argue, and that will also develop your relationship with the community of people who need the service. And I understand that you're building your model for doctors, but quite frankly, We think the model is more importantly, it should be built for the patient community and getting this direct information from the patient community. So let me suggest to you, Gary, that you open up a tick testing lab as well as part of this.
Well, let's go
In your
back.
free time.
Let's go back to one of your original questions about the entrepreneurial model. Here's the one thing that, if I could use my old age experience, that's really critical about entrepreneurial models, and it's called focus. And you can't be all things to all people, and you can't do everything. Now, we have significant research capabilities and everything. So and actually we were just doing an orthopedic based, because we are just now, I mean, what we're doing here is one of the first, and what the conversations we had this week is actually the first time we've come out of the closet with regard to what we're doing. We've stayed silent on this for a long period of time, but we did this little Shark Tank like thing that was orthopedic based. And we only had 14 minutes. I mean, it wasn't the right format to introduce our technology, but he suggested that we should only do one disease state. And even though I wasn't able to say, you don't know what you're talking about, because the tick-borne analysis gives us validation in urine. Sepsis gives us validation in blood. Orthopedics gives us validation in cerebral spinal fluid. So it's a big bite of the apple, but it creates the broad spectrum of what this technology can do in all these disease states. Now, if we start adding on to that, and we're trying to, because we're getting hammered with regard to the blood research, with regard to the infections that are generated by the internal mechanism of infection for burn patients. We have cardiac people wanting to, if you start doing too much, you really won't accomplish what you need. So even though it's a broad thing that we're doing, our expertise is what we do, and collaborating with the people who are gathering ticks is the value, I think, because we can't do it all. It's really, and again, and getting it to a point where we've really proven our case and hand it off to a bigger entity that can really blow it out to the country and to the rest of the world is really what we need to accomplish. That's the entrepreneurial model. And these days in the medical world, the fact of the matter is, most of the time, if you're dealing with medical devices, for instance, if you're a one-trick pony, you take it so far, and then you run into the door because you don't have the broad spectrum that J&J and Medtronic and all the other big dogs have, and you get locked out. So you hand off your product and say, take it forward, please. And if you believe in the contribution to the body of science, this sounds like I'm painting a halo, but I came out of retirement because retirement isn't my thing, if you truly believe in doing what you're doing because you want to help patients and contribute to the body of science, and yes, we'll make money as we go forward. But the fact of the matter is, when entrepreneurial companies dive deep into this and hand off to the big companies, that's how it happens. I'm going to use Medtronic as an example. I used to work for Medtronic years ago, and everybody thinks, and those from Medtronic listening to this are probably going to, but I don't care, wiggle. But everybody thought of Medtronic as this innovative company. They were a company of acquiring little technologies like this to allow them to be the giant that they are. That's why the entrepreneurial model you're talking about, that contribution of the entrepreneurs to the bigger entity and the well-being of patients, is important. Boy, I really got off on a tangent there, didn't I?
No, you didn't. I think you hit it right on its head, because I think the larger concern that I have, again, and we're a Lyme disease podcast, is that the work that you're doing is, I think, bigger than you even understand. Because at a very basic level, we don't even know what's being spit into us. And you're already to the point where you're identifying what's being spit into us, what the combination is, whether or not, the medications are going to work. And you were just speaking of antibiotics, but we should identify herbs. We should identify a lot of other factors that should be tested. And then, of course, the absorption rate, for example, of the individual person when they're being given the antibiotics, and it's just level after level after level of places where you're taking this. And I'm arguing to at the very basic level about what the pathogens are. We don't even have that. No less all these other levels of analysis that you guys are taking it to.
Well, and I'm gonna tell you, we are absolutely confident we have it And our methodology is going to be doing the science, because this is how I was brought up through the ranks, whether you're dealing with heart valves, defibrillators, pacemakers, vascular grafts. In the world that I grew up in, you do the science, you present the science for peer review. Otherwise, the insurance companies and the physicians aren't going to come on board unless you do the basic science. And so I think it's to the Lyme Society's benefit if we do this properly and bring everybody on board in the middle of the identification and the therapies that are needed to help these patients get over these horrific disease states.
I don't disagree with that, but one of the concerns that we have, and we interviewed Dr. McDonald, as I shared with you a couple of weeks ago, is that you have to find a place to publish your research, right? Mm-hmm. And the people who are the gatekeepers of these journals where you're publishing your research may or may not be interested in having your research made available to those journals. And then if your research is not made available to those journals, then they are not going to be part of the standard of care. And if that's not the part of the standard of care, then doctors are not going to use it because they don't want to be either sued or they don't want to find themselves before medical boards or licensing boards. So I agree with you that it will be wonderful, Gary, if you can take your research and you can make it available. I'm just not as confident as you are that you're going to be successful because of the political biases that you're going to have to overcome.
Well, yeah, and obviously, being somewhat experienced in this, I'm going to disagree because I'm one of these guys. I bite the tire and I hit the road as many times as I have to. But I take a contrarian view with regard to, because you touched upon something very interesting. But as we do the variety of disease states, if we just take it out of the broad spectrum of what we're doing, and we do sepsis and prosthetic joints, the credibility of the diagnostic capability will wash over with regard to the credibility. And the one thing that you run into, I got to be a little bit delicate here, because the one thing that you run into is if you have physicians who are studying a particular different methodology than you have, and you run against that brick wall, that, in this world, I've been doing this a long time, you recognize that and you go to those who understand, who don't have those biases, so to speak. There are enough scientific journals out there that when we get to publishing the science, not only behind tick-borne disease, but also the other disease states that we have, you're going to find that other companies are going to try to mimic what we're doing is really what it amounts to. But I believe that we will be able to get it out there, because there's not just one journal that will publish this information. There are many that will publish it, and we will find them. Because,
but Alex, I'm sorry, Gary, I agree with you. See, because your spectrum is broad, and because you're not just in the tick-borne arena, you are arguing that there's research. You're doing research on particular blood sampling and on urine sampling and on spinal fluids in arenas outside of the tick-borne illness. Once you get credibility in other places, then those folks who may have a political bias in the tick-borne arena are stuck with the results that you're getting in other places. So look, I'm excited about your approach. I get it, and I am excited about it, but I do want to caution our listeners because there's going to be a lot of excitement generated by what you guys are sharing right now. And some of that has to be, tempered by some of the political challenges that the organizations that you're trying to appeal to have prevented, doctors like Dr. Burascano and Dr. McDonald, who are now finally being perceived as being correct in their research and finally finding, publication avenues for research that they did 30 years ago. So, I think your timing is certainly better than theirs was, and your tools are different than their tools. And your approach, Gary, I love that you're in a number of different areas, not just tick-borne, but you should be aware that you're running into a political thicket.
Oh, actually, because I wasn't aware of the thicket until I accidentally ran like I said, we ran into this whole desire to address the unmet need of Lyme by having an orthopedic surgeon tell me a story about a friend of his who could no longer practice surgery, and he said, "You ought to apply this to Lyme." And that's when I went home, did the research, and the thicket became aware of it very quickly. As the old saying goes, the Lyme Wars. But and that's what we're out. That's what we're out to problem solve. Quite frankly, about we want to solve that problem of those thickets. We're going to cut them down.
So one of the burning questions I know every listener is probably thinking about this entire time is, when do you estimate that this testing will be brought to market for either patients to order on their own or through their doctor, so they can finally get the validation they need if they haven't had a positive diagnosis, or to confirm their suspicions about other tick-borne diseases beyond what their current diagnosis is? So do you have an estimated time frame that you think this will be available to the general public to really help change their lives, frankly?
Yes, I'll answer from my perspective, and then Dr. Shope, you can answer from your perspective. But we have work to do with regard to some CLIA validation, which we shouldn't take a long period of time. We want to work with Lyme knowledgeable doctors and do significant clinical studies that will be published. Our goal is to have peer-reviewed journal publications for Infectious Disease Society folks to peer review by the end of 2021. The answer to your question is I'm assuming that by the end of summer, going into the fall, we should be in a position whereby we can, quote unquote, charge patients for that test. But in the meantime, all the dollars spent, because I'm going to be out raising money to support, because these tests we run, every time we run a test, it costs thousands of dollars. So we have to have a multitude of samples in, and it's going to cost. Healthcare research, you don't realize how much money you have to spend to prove the points that we're talking about. So I'm going to be out raising investment money to support this. But then by the end of the year, I'm supposing that we can probably be in a position to charge based on CLIA validation issues. And we'll be continuing to doing the clinical studies going forward into 2022, but certainly in 2022, but I think at the end of 2021, if people want to pay to be tested, we'll be in a position to do that. I don't know, Dr. Shope, do you disagree with that timing? Because all that testing stuff is in your lap.
Yeah, that is, isn't it? No, I don't disagree with that general time frame. Absolutely, within a year is more than safe to say. I think by the end of this year, we will be in the position with the right amount of regulatory data to actually offer the test to the public, should they want to pay for it. And then, as my father mentioned, we'll be continuing to do the additional studies to do the the clinical proof that other physicians and people need in order to understand the test.
Yeah, and Rich, if I could address this to you, because I know this is some of the points of your conversation, because I come from a little bit of a bias that, again, that orthopedic broadcast we were on, everybody was the big .com bubble these days are point of care and bedside testing and et cetera. And we're a lab-based test because what we do is so complex. But I have this concern that if we don't do the science to properly inform infectious disease doctors, we can test a patient and we can say, this is what you have, but then what do you do with that information if you don't have a physician on board to prescribe the antibiotics to treat it? So I struggle with, how do we do this without getting individual patients wound up, without having an available treatment, by having credibility at the physician base? So we want to do this in concert with physicians and not just have a patient-driven information system. And you may or may not disagree with that, but that's something I struggle with.
Yeah, I do disagree with that, because I don't think the doctor should be driving the care model. I think the patient should be employing the doctor to establish the care model. And it actually has been in our experience, and again, we've interviewed almost 200 chronic Lyme disease patients at this point. And what we are seeing from the standpoint of the pattern of success when people are suffering from chronic Lyme disease is when they're empowered to take control of their care and they're building their team of professionals to assist them with their treatment model, rather than going to an infectious disease doctor, for example, and having that doctor, with the limited tools that they have, ultimately outline a care model for you. So yeah, I would, Gary, quite frankly, I think I would, with all due respect, disagree. I would prefer it be a patient-driven model rather than a doctor-driven model.
Yeah, no, these are good conversations to have, because obviously because we're reaching out, all good companies have market research, and this is great market research. But however, the only question I have, and I hear what you're saying, that I'm not negating that. My question is, if the patient initiating it, we get the test, but to treat the disease, if they have to get an antibiotic, the patient can't do that themselves. The physician has to write a script for the antibiotic. And that, I guess I'm trying to define my concerns is, if the patient doesn't have access to the antibiotic to treat it, then what are they going to do with that information?
But the patient does have access to an antibiotic. They go to their doctor, they give their doctor the test results that they have located themselves, and they talk with their doctor about what the proper treatment tools would be, one of which may be an antibiotic or particular type of antibiotic. So you do have access to antibiotics. You just have to get the approval of your medical partner to use that tool. So
Okay.
I do think it really should be patient-driven rather than doctor-driven. And part of the problem is, going back to the discussion we had a moment ago, which is doctors are often concerned about their licenses. They're often concerned about medical malpractice claims. They're concerned about a number of different challenges that are presented when the standard of care is established by politically driven people who are controlling access to medical journals. So the only way that we can overcome that challenge is by going directly to the patient community. And when you go directly to the patient community and they are now driving the care plan, then there's less risk to the physician, and there's a greater likelihood that I'm going to get the treatment that I need. I mean, I've had really bad experiences with doctors that I had long-term relationships with after I was bitten by a tick. And I now understand why the results were so bad. But quite frankly, that's what generated this podcast. It was my personal challenge with the doctors that I was working with, who I don't work with anymore, by the way, that drove me to working with Matt and working on this podcast. So again, I'm certainly happy to debate with you about this, and I'm not sure our listeners want to hear me and you debate about this anymore, but I strongly urge you to consider a patient-centered model rather than a doctor, and in particular, an infectious disease doctor, because we have not really had, in our experience, many patients work with infectious disease doctors and get a good result.
But isn't that, you've just described the thicket that exists, and I guess isn't the goal for us to change the thicket, whereby when a patient asks for a kit, they take it to the doctor recognizes it's a valid test and gives them the antibiotic. Isn't that what we're trying to get to eventually?
Ultimately,
I say antibiotic because obviously you're talking about things I know nothing about with regard to other therapy methodologies, and I don't want to go there, but I'm focused on antibiotics for
ultimately, Gary, I'm hoping and I'm praying that you folks are going to be able to give doctors the tests they need to come to the conclusions that they need to, because I think most doctors are afraid to make a clinical diagnosis and establish a care plan based on the clinical diagnosis because it creates risk. I understand that. I understand you don't go to medical school so that you can lose your license. You don't go to medical school so you can get sued. I get that. So we need some objective testing for these folks so that they feel comfortable with treating, because they're not comfortable treating with a clinical diagnosis. But we're a long way away from that. And what I'm arguing to the two of you is, you have gold that can help people and suffering from Lyme disease. And if we wait until you get through all of these processes that you're going to go through before it becomes a standard of care, many people are going to be sick chronically, and many people are going to die. So I apologize for the frustration, but that's
no. Well, give me 6 months. Give me 4 months. Let's do it that way. Give me 4 months to prove a couple of points. But and actually, we're actually opening up some doors with DoD, NIH, and some other hopefully CDC sometime. So to me, I think what we're obligated to do is to continue to drive as fast and as furious as we can with regard to impacting and knocking on all those doors with regard to credible information so that we do turn this ship around. All our little tugboats are going to turn this ship around so that the patients get the right therapy. In my view, we're going to put it out there, make it available as fast as we can. That's all I can promise you. But we're also going behind the scenes, make sure that we have science and trying to cut that thicket down a little bit so that the doctors are on board as well as the patients. That's my goal. That's, I hope, anyway.
And that's certainly our prayer. So, Matt, do you have anything else for these brilliant men that we've been blessed to interview today?
No, I just want to thank Carrie for introducing us to Alex and Gary. If it weren't for Carrie from Sam Spooney, we wouldn't have met Gary or Alex and been able to have this wonderful conversation and share this with our audience and provide hope for the future of the Lyme community. So I just want to thank both Gary and Alex and also Carrie for introducing us to you guys.
Well, thank you, Matt. Thank you, Rich. We appreciate the opportunity to speak to you.
Yeah, guys, thanks so much. This has been great again to know you guys more and continue to talk about this. So thanks for having us.
Let's continue these discussions. Absolutely.
Thank you for listening to the Tick Boot Camp interview with our guest, Dr. Alexander Shope, and his dad, Gary Shope. To our listeners, we have a call to action. First, if you'd like to learn more about untargeted testing for targeted Lyme disease treatment, please visit the webpage at csidx.com. Second, if you enjoyed this episode of the Tick Boot Camp podcast, please share with your friends by using the social media buttons you see at the bottom of the post. Third, Tick Boot Camp has created a Tick Bite blueprint that has been inspired by the information that has been provided by past podcast guests. We urge you to visit our website at www.tickbootcamp.com to view the blueprint. Please note we would appreciate any input or any improvements you would like to offer to us. Fourth, don't forget to subscribe to this podcast on iTunes, Google Play Music, or Spotify to get your automatic episode updates of our Tick Boot Camp podcast. And finally, we thank our community for your comments on our past podcast episodes. Please take a minute to leave us an honest review on iTunes, on Instagram, or on our website. We make it a point to read every single one of the reviews we get. Thank you for listening.